Hi and welcome to my OCR A-Level Biology revision session with me, Christine. So today's lesson I'm going to look at membrane structure. So all membranes are made up of the same basic structure, and that is a phospholipid. So the phospholipid has what's known as a hydrophilic phosphate head and a hydrophobic fatty acid tails. So every phospholipid has the same structure, and when it is placed into an aqueous solution, what will happen is those phospholipids will create what's known as a phospholipid bilayer. Two layers. Now, the phospholipid bilayer does this because the hydrophilic heads orientate themselves on the outside. They can form hydrogen bonds with water, and therefore they are going to orientate themselves on the outside, and the hydrophobic tails orientate themselves on the inside. So you end up with this hydrophobic region. Now that hydrophobic region creates what's known as a barrier. So it's a barrier that is going to prevent any large or polar molecules from crossing that hydrophobic region. So the plasma membrane acts as a barrier surrounding the cell. Well, inside the cell, we want to have little compartments. And what we want to do is have these compartments that have different roles to play. Now, if we think about the endoplasmic reticulum, the Golgi apparatus, the nucleus, these are all called membrane-bound organelles because they have this phospholipid bilayer surrounding them. So they're made of the same basic structure, which means they can fuse together and they can separate quite easily. And that's important when we look at protein synthesis for extracellular proteins. So whenever we describe the membrane structure, the biological membranes, we tend to use the term a fluid mosaic model. And the reason it's a mosaic model is because there are different proteins embedded in the bilayer. Now those proteins have different sizes, they have different shapes, and they are embedded in lots of different places, which gives the mosaic effect. Now we say it's a fluid mosaic model because the phospholipids and these proteins are free to move within the layer. They are fluid. So therefore we have a fluid mosaic model when we talk about the biological membranes because of the ability of them to move around. So they're not stuck in a position, they are able to move. So if we look at the proteins then, which are inside the membrane, we call the ones which are embedded and go through both layers intrinsic proteins. If they are inside the membrane, that phospholipid bilayer, they are known as intrinsic proteins. So carrier protein, channel protein, these are intrinsic proteins. Now we also have a glycoprotein. Now the glycoprotein is embedded in part of the membrane. Now if it is embedded in the membrane in the fact that it has these hydrophobic R groups which are going to interact with a hydrophobic core of the membrane, then it is classed as an intrinsic protein. Now the glycoprotein is an interesting one because the glycoprotein has that name because the glyco talks about the carbohydrate chain which is attached to the protein. So the membrane has a very specific role to act as a barrier, be selectively permeable, only allowing some things in and not all things, and therefore it has to have other parts to it which are needed for the cell to function the way it does. So the glycoproteins are there for cell adhesion, adjacent cells sticking together, but they're also there and act as receptors. So a glycoprotein can act as a receptor. Now, whenever we show these proteins, they can either be glycoproteins on the outside or they can be proteins which are embedded all the way through. Now, the whole point in these glycoproteins, these receptors, is to signal different cells. So what you have when you get to module 5, you will learn about the effects of adrenaline. You'll learn about the effects of acetylcholine, noradrenaline. You'll learn about insulin and glucagon. So it's important that you note that the proteins that are in the plasma membrane have got a role to play in cell signaling, the communication between the cells within the body. Now, another thing that we have when we look at the structure of the cell membrane is what's known as a glycolipid. So just like the glycoprotein, the glyco is talking about a carbohydrate chain attached this time to the lipid. Hence, it's called a glycolipid. Now, the glycolipids are for cell recognition. So they can be called antigens. And those antigens are either self-antigens, so your immune system recognizes your own cells as being self cells and therefore will not attack, or these antigens can be recognized as non-self, and that is where the immune system will be able to then have a role to play to counteract this pathogen, whatever is inside. So do check out my video on the communicable diseases where I cover antigens in more detail. So we have these intrinsic proteins, we have these glycolipids, we also have peripheral proteins, or what's known as extrinsic proteins. So if we have a protein which sits and interacts on the outside and interacts with the polar heads of either the phospholipids, or it interacts with the outside of the intrinsic proteins, they are called extrinsic proteins. Now, that's because they have hydrophilic R groups. They're hydrophilic, which means that they form hydrogen bonds. And because they can form hydrogen bonds with water, they sit on the outside of the membrane. So they're not embedded inside the membrane, they're sitting on the outside, and they can be either on the outside, which is inside the cell where the cytoplasm is, or they might be embedded at the extracellular fluid side and again interacting with the membrane there. They are called extrinsic proteins because they're on the outside of the membrane. So proteins have got lots of roles to play, and as we said before, there are proteins embedded in the membranes. And if we look at organelles, specifically this one, right now we're inside the mitochondrion. Inside the mitochondrion, there are proteins which are needed for sites of chemical reactions. So when you look at oxidative phosphorylation in module 5 and respiration, you'll be learning about the electron transport chain and an enzyme called ATP synthase, which is found inside the cristae. Now, these proteins need to be in the right position in the membrane for the role in which they have to play for these chemical reactions. So proteins inside the membrane have got lots of different roles to play, and it just depends where they need to be. So you can see here I have a few which are intrinsic proteins, and I also have an extrinsic protein, which is all necessary as part of the electron transport chain. So the membrane then, we know is fluid, we know things can move around. So what we need is a way in which we can stabilize the membrane, and we do that through the use of cholesterol. So cholesterol is another type of lipid which is able to embed into the membrane. Now, what that does is that stabilizes the membrane by decreasing the fluidity at high temperatures and preventing the phospholipids grouping too closely together and crystallizing at low temperatures. So it stabilizes the membrane, making it less fluid or more fluid, depending on what is needed. And that comes into play if we think about the factors that affect the membrane structure. So if we look at temperature, When we have low temperatures, the membranes are intact, and they're intact because cholesterol actually increases the fluidity at low temperatures. And that's important to allow for molecules to still move across into or out of the cell. So do check out my video on transport across membranes, where I'll talk about the different movements. But the cholesterol is necessary to ensure at low temperatures those phospholipids don't pack together too closely and therefore prevent molecules from moving across. Now, if we increase the temperature, what we would find is there would be more kinetic energy. So if we have an increase in temperature, we can see that there's more kinetic energy. If there's more kinetic energy, there's going to be more movement of phospholipids. If they are moving around more, they're therefore going to become more leaky. Now remember, cholesterol decreases the fluidity at high temperatures. We don't want the membrane to become too fluid because we don't want the cytoplasm or a pigment to leak out. So whenever we do this as a practical situation, we tend to look at things like beetroot. So beetroot has that pigment inside, the purple colour, and we can look at how leaky the beetroot becomes. So therefore the membrane of the beetroot, its permeability, based on what happens when we increase temperature. Now, if we increase the temperature, if we've got more kinetic energy, if we've got more movement of these phospholipids, that membrane is going to become more permeable. These contents are going to leak out. And you can see that in my graph, in the fact that up until the 20 degrees C, the membrane wasn't really changing the amount of pigment that had been leaked out. Once we got to 30 degrees and 40, then 50, then 60, we can see a gradual increase between 30 to 40 degrees, but then a much higher increase between 40 and 50, and then between 50 to 60, we have this exponential increase of the movement of our pigment. Now that's because as the temperature gets higher, not only are the phospholipids and proteins moving around more, but you also have the fact that their structure, those proteins, are going to denature. The high temperature is going to cause more movement, and therefore that movement breaks those interactions within that protein. That again will increase the permeability and cause more leakage out of the cell. So it's important that you note that temperature is a factor that's going to affect the structure of the membrane. And the other one you need to know about is to do with the solvents. So organic solvents are less polar than water. Because they're less polar than water, what that means is they will be able to dissolve membranes because they can disrupt the cells by slotting in to the phospholipid bilayer. So if we look at ethanol, for example, ethanol we know is an alcohol, therefore it has a hydroxyl group, so it has a hydrophilic part to it, but the majority of the ethanol is hydrophobic. That means it can insert itself into that phospholipid bilayer. So because it can insert itself into that phospholipid bilayer, it's going to push the phospholipids apart. It's going to disrupt the membrane and therefore make them more leaky. And that's why we tend to use things like alcohol in our antiseptic wipes or hand gels, because we're not just talking about the membranes in our cells, we're also talking about the membranes in prokaryotic cells. So alcohol, organic solvents, because of them being less polar than water, because they can embed themselves into the hydrophobic region of the membrane, they therefore disrupt the membrane, and that causes the cell to become more leaky. The higher the concentration, the more leakage you will get. Now, it's important to also remember that the ethanol will also have an effect on the protein. So the proteins as well, not only fitting into the phospholipid bilayer, but the proteins will also start to change shape because of the concentration of ethanol increasing. So the structure of the membrane is affected by the solvent fitting and slotting in between. Now, with an increase in the fluidity of the membrane, we're going to make it more permeable. More permeable, the substance inside is going to leak out more. So what we tend to do is we tend to use a colorimeter to observe the changes that are happening. So if you haven't already, do check out my video on quantitative data collection. So if you like this video, then please do click the like button and subscribe to my channel. Also, do check out my revision platform, www.aiqchat.com, to help you with your revision.